Namaste to all. Welcome to the next session on molecular biology and our DNA technology. In this session, we are going to see about DNA replication basics as well as we are going to see about enzymes which are important for DNA replication. So, if you see the enzyme's role, they're able to perform two functions. The first thing is autocatalysis, that is DNA to DNA, that is replication, and heterocatalysis, that is DNA to RNA, that is transcription. So, autocatalysis is very much important, that is, DNA replication occurs on its own. But for occurring this, they require many enzymes for its complex multi step process of DNA replication. So if we see the semi-conservative method of replication, we know that the DNA has two strands, which are 3' to 5' and 5' to 3'. They are the parental strand. During the process of replication, these strands, they separate and become like a loop, or they form the bubble. In this bubble only, the replication process they start. After the replication process is done, we are going to get 3' to 5' and 5' to 3' as a daughter strand. And these two strands, after this process, they get separated and can be used for the next cell. So enzymes used during DNA replication. Helicase. It helps in unwinding the DNA coil. It open up or unwind the DNA helix. This happens by breaking the hydrogen bond between adenine and thymine and cytosine and guanine. Topoisomerase. It actually removes the tension which is coming near to the nucleosome. So during the unwinding of the DNA helix, and cuts, rotates, and reseal the strands. Single-stranded stabilizing protein or binding protein. This helps or makes the strand stress-free and stabilizes the strand during the replication process. Ligase. Ligase helps in joining the DNA fragments. Basically, it helps for the joining of the Okazaki fragments. DNA polymerase 1. It helps in the removal of primer. It helps in filling up of the gap also. Primase, it helps in the primer synthesis. It is actually DNA dependent RNA polymerase which is required at this particular step. Primer, if you see, they are small segment of RNA nucleotides containing 20 to 25 bases. If you see the primase in the 3' to 5' strand or 5' to 3' strand, usually they join at the starting position and synthesize the RNA primer. After synthesizing the RNA primer, the DNA polymerase, it helps to synthesize few strands or few bases as an initial sequence. DNA polymerase 3. This helps as an important enzyme in DNA synthesis in replication. It works as dimer and simultaneously synthesizes in both the strands, that is 5' to 3' and 3' to 5'. In eukaryotes, especially, they have these as three subunits. The first subunit is alpha subunit, which is catalytic unit for the initiation, for the formation of the bonds between the nucleotides. Epsilon subunit, that is, synthesizes the leading strand and helps in proofreading. And delta subunit, they synthesizes the lagging strand. Beta subunit, it acts as a sliding clamp. Phosphorylase. It helps in addition of phosphate, that is, adding to the nucleic acid, that is monophosphate. It becomes activated and becomes as nucleic acid triphosphate. And this activated form is actually attached during the process, and then it is helpful for the making of the bases. Nucleases, that is, helps in proofreading. I hope that in this video you have got an idea that what are the different enzymes which plays an important role during DNA replication. See you in the next session with replication, origin of replication, prokaryotic replication, and eukaryotic replication. Thank you and have a good day.